Overview of introduction of nucleus and types of cells and the components of the nucleus, the nuclear envelope, and what is the nucleoplasm, and what is the nucleoplasm, and what is the nucleolus, what is the nucleolus, and chromatin and molecular molecular weight. And then what is the chromatins, how to use for molecular structure of the chromatins. These are things are the most important for the nucleus. And the next one is the nucleus membrane. Nucleus is the Latin word. This one is the Latin word. And next one, the meaning of Neil. The meaning of the Neil, the main things of the nucleus is called the, one second, happening. The nucleus, nucleus is the Latin word meaning of the kernel scientist. It is a control center. The nucleus is called the control center and control center of the cell. It was first cell organelle to be discovered by. In this one is the discovered it membrane bound organelle found in the eukaryotic cell. Nucleus is the main functions are to maintain the integrity of genes, to control the activities of the cell by regulation of gene expressions. These are all involvement of the nucleus is there. Okay, what is the nucleus? Nucleus is nucleus, this one is the meaning of Latin word. Kernel scientists to explain that one. And nucleus, it is a central control center of the cell. All the main important of the control of the cell is called the nucleus. It was the first cell organelle to be discovered. It is a membrane bound organelle found in eukaryotic cell. Main functions are to maintain the integrity of genes, to control the activities of the cell by regulating the gene expressions also. And the nucleus function, it is a store the cell hereditary material. Main is the it is a store the hereditary function and or DNA. Hereditary functions are DNA is the most important in the nucleus. And another thing, site of the replication also. Site of a replication also. Site of replication to mRNA. And ribosomal formation also present. Ribosomal formation and next one, nucleolus RNA and proteins required for ribosomal synthesis. It coordinates the cell activities, which include growth, intermediately metabolism, the protein synthesis, and the reproduction of the cell division by regulating gene expressions. All are the under control the nucleus also. These are the function of the nucleus. Coming to the structure, this is not move. What happened? Another one is the that's nucleus structure is called the average diameter of nucleus 6 millimeters, which occupy around 10% of the total cell volume. What is the how much of the occupy the total cell volume? The nucleus also occupy the only for the 10% of the cell volume, and the content of the nucleus. One second. Nucleus. Here. Notes. The total cell volume, the nucleus to occupy 10%. Only 10% of the nucleus is the occupation are there. And the content of the nucleus are enclosed by a complex nuclear envelope. The nuclear envelope also main important. In this one here, nucleus, main under nucleus you can observe chromatins, or nucleoplasm, nucleolus also present. Nucleolus is the concentrated area of chromatin, RNA, and proteins. These are the nucleolus areas. And next one, the nuclear envelope, that is the nuclear envelope completely enclose the nucleus and separate the cell's genetic material from the surrounding cytoplasm. The services surviving as a barrier to prevent macromolecules from diffusing freely between the nucleoplasm and the cytoplasm. This is also perinuclear envelope is there. This one is called the perinuclear envelope is present. Nuclear membrane also present. The nuclear membrane, this nuclear membrane, nuclear membrane also is called the perinuclear membrane. Nuclear membrane is called the perinuclear membrane. This one is the space and it continuous with the lumen also. Another one, nuclear core also present. It is complex in the nuclear membrane that enable to the traffic of molecule between the nucleus and surroundings. This nuclear envelope, lamellas and present. Sometimes the lamina and nucleolus both are the present in this one processing. Where the interface chromosome it present in a healthy extended nucleoprotein fibers. This is called the chromatin. Chromatin is the complex of DNA and proteins that makes up the chromosome. Each unreplicated chromosome contain a single constitute DNA molecules. The mitotic process chromosomes represent a highly condensed structures are present. This is the chromosome. Nucleolus also very important in this one. Nucleolus is the discrete, densely stained structure found in nucleus. It is a nuclear inclusion that is not surrounded by the membrane present in cells that are actively synthesizing proteins. Its size depend on metabolic activity of cell average size 5.5 millimicrons. The average of cells is microns in the diameter of nucleus. It is mainly associated with the synthesis of rRNA. The nucleolus mostly importance for the rRNA. Nucleoplasm. Nucleoplasm is the protoplasm. Nucleoplasm is the protoplasm within the uh, within the within the nuclear envelope. It consists of nuclear envelope. It consists of nucleus envelope. It consists of a nuclear matrix and various types of particulars highly viscous and liquid, highly viscous and liquid with scaffold chromosomes. With scaffold chromosomes. PPT. Oh, yes. This one is the mostly here involvement of this one is very particular, and next one is called mm, one second, I'll share my PPT. Yeah, this is the discovery of the nucleus. That four stages also you can observe the nucleus. That is the growth. Who is the which one? Which scientist to discover? See this total nucleus in this one. In the nucleus for this, you can identify observe the introduction of nucleus, nuclear envelopes, and transport of the transport through nucleus, transport through nucleus, and how to nuclear membrane is present. These are the most important for the nucleus and introduction of this one. And the what is the nucleus main important now, except the mature mammalian RBC, where the nucleus is present, all living cells nucleus is present, except where the nucleus is absent, where the human mammalian cells, human mammalian cells, this one is absent, nucleus is absent, and the next one RBC cells also. Where the RBC cells are there, in that RBC cells also, nucleus is absent. Where the sieve tubes or sieve elements, sieve cells are there in plants, in the starting cell, this nucleus is absent. In the sieve cells also, nucleus is absent. Next one, the size and shape of the nucleus. Usually, the shape of nucleus is a spherical in shape. That one is called the spherical in shape, or sometimes oval, but can be discoid shape, irregular shape, typically occupy about to the total cell volume, 10% of volume. This is the introduction. What is the ultrastructure? Ultrastructure of the nuclear, where the interface nucleus consists of four main components. What are they? First one is the nuclear envelope, nuclear pore, nucleoplasm, nucleolus, chromatin network. These are the most important. The first one is the nuclear envelope, karyotheca, a double membrane structure, outer and inner separate by a perinuclear pore. The outer membrane is often continuous with the rough endoplasmic reticulum. Rough endoplasmic reticulum. The main important of this one, nuclear pores, circular opening, approximately 120 nanometer in diameter, formed by the fusion of the two membranes. They regulate the exchange of materials, RNA and proteins between the nucleus and cytoplasm. The nucleoplasm or another name is called karyoplasm. a transport semi-solid granules, ground substance containing water, mineral, protein proteins and enzymes for the DNA and RNA synthesis. Coming to the next one, nucleolus, a dense spherical membrane, then membrane less body rich in RNA and proteins. It is the site of the ribosomal RNA, rRNA, where the nucleolus layer, rRNA synthesis and ribosome assemble. The chromatin network, thread-like structure composed of DNA and histone proteins during cell division. These condensed into chromatin. Another one is the functions of the nucleus. Main function genetic information stores DNA, the hereditary blueprint of the organism. Main important hereditary of the DNA and blueprint of the organisms. Control central is regulation of cell growth, metabolism, and protein synthesis by the controlling gene expressions. Gene expressions. Cell division, the replication of DNA to ensure genetic continuity during the mitosis and meiosis. And next one, ribosome production. The nucleolus production to the subunits of the ribosome required of the protein translation. This is the function of the nucleus. And another thing, what happened? It is not move. I'll stop sharing. Again, I'll start. It's very stuck. And the main important of the function, nucleolus is the genetic information also most important. Where the genetic information to carry the that one also that is called very information are there. And this is the main see this is the total structure of the nucleus. This one is the membrane, nuclear membrane, outer membrane and inner membrane. See, this is the nucleolus outer membrane. This part is called inner membrane. In between empty place is there. This is a perinuclear space. Outer and inner, in between the space, perinuclear space. This is called the nuclear lamella. And this one is a nuclear pore complex, nuclear envelopment and atomy, and these are the ribosomes. Also called nuclear envelope doubled membrane also there. One is the porous and nuclear lamina also present. Here, main important the lamina protein network that stabilized of the nucleolus and found just inside the nuclear membrane. What are the main function? The control of the central of the cell and contain DNA genetic material also to contain that genetic material is called the DNA. Direct to protein synthesis. The ribosomes to protein synthesis to this one and contain genetic information also. And another important character, nucleolus. This is the small and dark region of nucleolus, place where the ribosomal component are, are made up of. The other nodes of the nucleus, DNA is the found in the in the formation of chromatins. The thin and uncoiled chromosomes. Thin and uncoiled chromosomes only form when their cells is absent about to divide. That is the other corners of this one. What is the made in the nuclear? Nuclear made up of the nucleolus, made up of the ribosomes and lipids, carbohydrates, ATPs, and these are things are the most important to the lipids. And these both are the great job for the lipid metabolisms. This one is the nucleus. Now, already in the Sunday class also, we will discuss about to the nucleus. Coming to the next one, nucleic acids. What are the nucleic acids? How many types of nucleic acids? These are things now we will discuss about. Inside the nucleus, two nucleic acids are present. One is the RNA, another one is called DNA. See, nucleic acid naturally occurring in the chemical pattern. In the nucleolus, four parts main important. Nuclear membrane, outer membrane, inner membrane, in between nuclear intramembrane or nuclear membrane in between, in outer and inner. These are the intermediate like structures are present. Another side, nuclear membrane, and inside thread-like structures are present. That one name is called chromosomes or chromatins or genes. Each and everything, the nucleolus chromatin-like structures are there. That chromatins is called the chromosomes. Nucleotide, chromosomes, nucleolus. These are the inside the you can observe nuclear envelope only two nuclear envelope and nuclear envelope and functions of nuclear. Inside the nucleoplasm, the chemical-like structures are present. That name is called nucleic acids. Here, nucleic acids naturally occurring chemical compound that serves as that one main function formation during the service, the carrying from information molecule for the cell, and that direct process in the process of protein synthesis, thereby determining the kind the inherent characteristics of the every living things. Nucleic acids are further defined by their ability to be broke down to broke down to the yield. The phosphoric acid, sugars, and matrix from organic bases. Yeah, organic bases meaning purine and pyrimidids. The two main classes of nucleic acids are there. One is the DNA, another one is called RNA. DNA is called deoxyribonucleic acid. RNA is the ribonucleic acid. DNA is the master blueprint of the life, and this is the constitute the genetic material in free living organisms. The free living organisms to found, this is the best viral, this is the most important viruses. RNA is the genetic information or genetic material from of content information that genetic material of the orient viruses or but it is also found in all length, all living cells, where it plays an important role of the certain process, such like the protein synthesis, building bodies. Main basic structure of nucleic acids. Nucleic acids are polynucleotide, that is the long chain like molecules composed of a series of a identifiable, nearly, identi- identical building blocks of called nucleotides. Each nucleotide consists of a nitrogen bases. These nitrogen bases are possible for the 4 to 5 types of nitrogen bases: adenine, guanine, cytosine, and thymine, and uracil. The adenine, guanine, thymine, and cytosine are uracil. The adenine, the each nucleic acid contained for a five the range of nucleic acid possible for the nitrogen mixing. Yeah. Now, each nucleic acid contain four, and other one is the purines, cytosine, thymine, uracil. Here present, G are categorized. Guanine, guanine, uracil, uracil, and thymine, and called that is called the pyrimidines. All nucleic acid contain the base A, B, C, and yeah, adenine, guanine, adenine, cytosine, guanine, and the divided USA is divided DNA is the next one, each nucleic acids, pento sugars are present. That is the deoxyribose. That's where DNA pentosugars at the deoxy, deoxy, deoxyribonucleic acids. The second one is the RNA, ribonucleic acid, while U is found in RNA. DNA is not heard. Hmm. Next one is that the pentose sugars, DNA differ from the sugars in RNA by the absence of hydroxyl group. The this here 2' carbon for the thinking, 2' two from the pentose sugars differ from the next one, the sugars, pentose sugars of DNA differ from the sugars in RNA by the absence of hydroxyl group. The second carbon of sugar ring download without without an without next one, pentose sugars. The phosphate group contains successive sugars reduced by the 5' hydroxyl group. Hydroxyl group of next sugar in the chain. This biosynthesis, nucleotides are synthesized from radially available, that is called the precursor in the cell. The ribose pentophosphate portion of the both purines and pyrimidines nucleotides is the synthesized from glucose via the pentose phosphate pathway is present in the sixth atom of the pyrimidine cycle. This is the synthesized first, subsequently to attach the ribosome pentose phosphate. These two rings of the proteins are synthesized by within attached to the ribosome phosphate. Phosphate during the assembly of the academic or granite and beside, this one is the beside finally the specialized enzyme is called during the assemble adenine and guanine nucleotides. Here main important sugars, pentose, nitrogen bases, that is a nucleotide. Where the sugars, pentose, phosphates is absent. Phosphate is absent, only sugars and nitrogen bases are there, nucleoside. Now, in both cases, the end product of the nucleotide carrying a phosphate attached to the 5' carbon on the sugar, finally a specialized enzyme to involvement in a special enzyme that is called the special enzyme kinases enzyme. Add a uh, add two phosphate group using adenosine triphosphate and the phosphate donor to form ribonucleotide triphosphate, the immediate precursor of RNA. For the DNA, 2 hydroxyl group is removed. For the ribonucleotide